Hello. In today's video, we shall learn in detail about the plant tissue system. By the end of this video, you'll have a clear understanding about the epidermal tissue system, the ground tissue system, and the vascular tissue system, along with the different types of vascular bundles. Let us start with the introduction to tissue system. A group of cells performing a specific function is known as tissue. Different types of tissues in a plant that perform similar basic function is known as tissue system. It is a functional unit derived from protoderm. Protoderm is a thin outer layer of meristematic tissue in vascular plants. Tissue system can be categorized into three types: the epidermal tissue system, the ground or fundamental tissue system, and the vascular tissue system. Let us understand the epidermal tissue system. It consists of a single outermost layer of parenchymatous cells, compactly arranged with no intercellular spaces. It is a protective layer which extends over the entire surface of the plant body. It contains cytoplasm, nucleus, and cell sap. During primary growth, the covering of plant body is known as epidermis. In roots, it is known as epiblemma or piliferous layer or rhizodermis. During secondary growth, the epidermis may be replaced by periderm. In the leaves, the epidermis possess numerous minute openings called stomata for exchange of gases and transpiration. The leaves possess both upper and lower epidermis. In xerophytes, there is a waxy deposition of cutin on the outer walls of the leaf epidermis. This cutinized layer is known as cuticle, which checks excessive loss of water through evaporation. The waxy coating acts as a moisture barrier and protects the plant from intense heat of sunlight. The epiblemma possess unicellular root hairs to increase the absorbing surface area for absorption of water and mineral salts from the soil. The functions of the epidermis are as follows. The epidermis acts as a protective tissue for the entire plant body. Due to the presence of cuticle, the epidermis checks excessive loss of water through evaporation. It helps to regulate gaseous exchange like oxygen and carbon dioxide between the leaves and the atmosphere. Root hairs present in the epiblemma help in absorption of water and minerals from the soil. Epidermis protects the plant from excessive heat and cold. It also protects the plant from infection by parasitic fungi and bacteria, and also from the attack of herbivorous animals. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, let us look into the structure of stomata. Stomata are the tiny openings present in the epidermal layer of the leaves. They play an important role in gaseous exchange, transpiration, and photosynthesis. The stomata consists of minute pores called stoma, surrounded by a pair of guard cells. The guard cells are living, have a cytoplasmic lining, contains a nucleus, central vacuole, and a number of chloroplasts. The inner walls are thicker and outer walls are thinner. The subsidiary or accessory cells surround the guard cell. The stomata open and close according to the turgidity of the guard cells. When the guard cells are turgid, they expand, resulting in the opening of the stomata. When the guard cells lose water, they become flaccid, leading to the closure of stomata. Functions of stomata are: they help in gaseous exchange between the plant and the atmosphere. For easy diffusion of oxygen and carbon dioxide, each stoma opens internally into a respiratory cavity which communicates with the intercellular spaces and air cavities. Stomata helps in removing excess of water through transpiration. The stomata maintains moisture balance according to weather conditions by opening and closing of stoma. It facilitates carbon dioxide uptake and release of oxygen during photosynthesis. Xerophytic plants have stomata which are embedded deeply in the leaves and are not exposed to sunlight directly. These sunken stomata are an adaptation to check excessive transpiration and water loss. Next, we shall learn in detail about the ground of fundamental tissue system. Ground tissue includes all tissues except epidermal tissues and vascular tissues. The ground tissue system is derived from a ground tissue meristem of the embryonic region. The system forms the main bulk of the plant body and includes the cortex, endodermis, pericycle, and the pith. Ground tissue is heterogeneous in nature, consisting of different types of cells which are adapted to carry on different functions. The different simple tissues are parenchyma, collenchyma, and sclerenchyma, of which parenchyma is the most abundant. The parenchyma cells are thin-walled, living cells, mainly present in the cortex, pericycle, pith, and medullary rays in primary stem and root. Collenchyma cells are also living cells with thickenings of pectin present at the corners which give extra mechanical and structural support to the plant. Sclerenchyma are thick, lignified, dead cells whose main function is to give structural support. Ground tissue called the cortex lies between the epidermis and the pericycle. In dicot stems, it is differentiated into hypodermis, general cortex, and endodermis. The cortical cells may contain chloroplasts, starch, crystals, etc. The hypodermis contains a few layers of collenchyma or sclerenchyma cells. General cortex is made up of thin-walled parenchyma cells. The endodermis is the innermost layer of the cortex. It is single-layered and may contain starch grains and hence also known as starch sheath. The endodermis shows presence of waxy substance suberin in the form of strips on the radial and transverse walls. These are known as Casparian strips. In monocot stems, there is no differentiation of cortex. It is made up of many layers of thin-walled parenchyma. It shows a distinct circular layer of endodermis, which is adjacent to the pericycle and is composed of closely packed cells. In general, the cortex surrounds the vascular cylinder and the pericycle. The ground tissues outside the stele are known as extrastellar ground tissue, and those inside the stele are known as intrastellar ground tissue. Example, pith. Cortex is primarily a protective layer. Its other functions are manufacture of food, storage of food, and giving mechanical support to the plant. Pericycle lies inside the endodermis and is the outermost part of the stele of plants. In dicot stems, it forms a ring around the vascular bundle. Pericycle may be made of both parenchyma and sclerenchyma cells. Patches of sclerenchyma associated with the phloem or bast of the vascular bundle forms a cap known as hard bast. In roots, there is only a single layer of pericycle made up of thin-walled, barrel-shaped cells. In all stems, the pericycle is the seat of origin of adventitious roots. In roots, pericycle initiates the formation of lateral roots, vascular cambium, and cork cambium. In dicot roots, the pericycle gives strength to the roots and provides protection to the vascular bundle or the stele. The cells of the pericycle in dicot roots become meristematic, divide, and give rise to the cork cambium or phellogen. Pericycle may show the presence of laticiferous cells and secretory cells. The primary meristem gives rise to the lateral roots. During secondary growth, the pericycle forms the vascular cambium and cork cambium. Next are the pith and medullary rays. The pith or medulla is usually made up of parenchyma cells with intercellular spaces. It occupies the central region of the stem and root. Pith develops from the inner portion of the ground tissue system. Pith cells may possess tannin and crystals. In dicot stem, pith is small. In monocot stem, since the vascular bundles are scattered, the pith cannot be identified. In monocot root, the pith is large and well developed. In dicot root, pith may be lacking. In dicot stems, pith is seen in the form of narrow parenchymatous strips in between the vascular bundles. They are known as pith rays or medullary rays. These rays serve as storage tissue. Pith cells, which are sclerenchymatous, give mechanical strength to the plant. Medullary rays transport food and water. Prior to secondary growth, the cells of medullary rays become meristematic and give rise to interfascicular cambium. Next, let us understand the vascular tissue system. The vascular tissue system consists of vascular bundles distributed in the stele. The stele consists of pericycle, vascular bundles, medullary rays, and pith. The vascular tissue system is made up of complex tissues, xylem and phloem, which constitute the vascular bundle. Vascular bundles originate from the procambium of the apical meristem. The xylem helps in the conduction of water and mineral nutrients from the roots to the leaves. The phloem helps to transport prepared food materials from the leaves to other parts of the plant. In stems and roots, the xylem lies closer to the pith, while phloem lies towards the exterior part. In dicot stems, meristematic cells called cambium are present in between the xylem and phloem. Cambium is absent in monocot stem. The vascular bundle in dicot stem and root are arranged in a circular ring. In monocot stems, the vascular bundles are scattered in the ground tissue. The vascular cambium gives rise to secondary xylem and secondary phloem and thus increases the girth of the plant. The cork cambium gives rise to thickened cork cells. These cells protect the plant and also reduces water loss. Now, let us study about the elements of a vascular bundle. The vascular bundle of a dicot stem shows three well-defined tissues, namely xylem or wood, phloem or bast, and cambium. Xylem lies towards the center and consists of vessels, tracheids, xylem fibers, and xylem parenchyma. The vessels may be spiral, scalariform, annular, reticulate, and pitted. The vessels and tracheids of xylem are responsible for the conduction of water and minerals from the roots to the various parts of the plant. Xylem parenchyma help in storage of food, while xylem fibers give rigidity. Among the different elements of xylem, only wood parenchyma is living, while the other elements are dead and lignified and therefore give mechanical support to the plant. The first formed xylem are the protoxylem. Annual and spiral thickenings are the characteristics of protoxylem elements. Xylem is endarc in stem, that is, protoxylem with vessels having smaller cavities are towards the center, while metaxylem, which are formed later with bigger reticulate and pitted vessels, are away from the center. Its development is centrifugal. In roots, xylem is exarch, with protoxylem towards the circumference and metaxylem towards the center. Its development is centripetal. Phloem lies towards the periphery and consists of sieve tubes, companion cells, and phloem parenchyma. They are all made up of cellulose and are living. Bast fibers are generally absent in primary phloem. It may show patches of pericyclic sclerenchyma called hard bast. Phloem helps in translocation of prepared food from leaves to other parts of the plant. Protophloem are the first formed phloem found on the outer portion of phloem, and it consists of narrow sieve tubes. The metaphloem are formed later and are complex in structure. They are found on the inner portion of phloem and they are formed of bigger sieve tubes. In plants having secondary growth, the primary phloem is of short duration and gets crushed. The leaves of gymnosperms possess, in addition to normal tissues, another conducting tissue called transfusion tissue. They are made up of short tracheid-like cells which have thin cellulose wall with bordered pits but is devoid of protoplast. In between the xylem and phloem, thin-walled, rectangular, elongated, meristematic cells may be present. These cells are called cambium. It is responsible for secondary growth and brings about increase in girth or thickness of the plant body. Finally, let us understand the different types of vascular bundles. According to the arrangement of xylem and phloem, the vascular bundles are of the following types: radial vascular bundle and conjoint vascular bundle. Conjoint vascular bundle may be open collateral vascular bundle or closed collateral vascular bundle. Next is bicollateral vascular bundle. And third is concentric vascular bundle, which may be amphicribal or hadrocentric and amphivessel or leptocentric. Let us learn about the different types of vascular bundles with the help of diagram. Radial vascular bundle. Primary xylem and primary phloem strands are separated from each other by non-vascular tissues and they lie on different radii alternating with each other. Conjoint vascular bundle. In this type of vascular bundle, xylem and phloem combine into one bundle. They are of different types. Collateral vascular bundle. The xylem and phloem lie together on the same radius. Strands of phloem is present external to the strand of xylem. Cambium may be present or absent between the xylem and phloem. When cambium is absent in between the xylem and phloem, it is said to be closed collateral vascular bundle. There is no secondary growth. Example, monocot stems. When in a collateral bundle, the cambium is present, it is said to be open collateral vascular bundle. Example, dicot stems. Bicollateral vascular bundle. In a collateral bundle, both phloem and cambium occur twice. Phloem is situated on the peripheral and inner side of xylem. Outer cambium is present between the peripheral phloem and xylem, and inner cambium is present between the inner phloem and xylem. The sequence of vascular tissue in bicollateral vascular bundle is outer phloem, outer cambium, xylem, inner cambium and inner phloem as clearly seen in the diagram. Concentric vascular bundle. A vascular bundle in which one type of vascular tissue surrounds the other. When xylem lies in the center and is surrounded by phloem, the concentric vascular bundle is known as amphicribal vascular bundle or hadrocentric vascular bundle. When phloem lies in the center and is surrounded by xylem, the concentric vascular bundle is known as amphivessel vascular bundle or leptocentric vascular bundle. So today we have learnt in detail about epidermal tissue system, ground or fundamental tissue system and vascular tissue system. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comment section below. I'll see you there. Goodbye. All the best.